I am Dr. Khokon Kumar Dotto. My Christian name is Dr. David Boone. I am currently serving as the chairman and assistant professor for the Department of Biochemistry and Molecular Biology in Bangabandhu Sheikh Mujibur Rahman Science and Technology University in Gopalganj, Bangladesh. The title of my discussion is Diabetes Medication Metformin Lowers Thioredoxin-Interacting Protein TXNIP in insulin-producing pancreatic beta cells and in vascular endothelial cells. Metformin is a diabetes drug. Metformin is approved by the US Food and Drug Administration (FDA). The doctors usually prescribe metformin as an adjunct to diet, exercise, and lifestyle changes such as weight loss for glycemic or sugar control in diabetes patients. Metformin is known to decrease glucose production by the liver, to decrease glucose uptake from the gastrointestinal tract, to increase insulin sensitivity of the target cells and others. Here the discussions are mainly concentrated on a published research article where metformin was reported to suppress the level of thyrodoxine-interacting protein TXNIP in pancreatic beta cells, which work as the factory for insulin production. Mayan Shakid et al. Published an article in December 2011 in the Journal of PLoS One entitled AMP activated protein kinase AMPK mediates nutrient regulation of thioredoxin-interacting protein TXNIP in pancreatic beta cells. The group reported that AMPK acts as an important regulator of TXNIP transcription in pancreatic beta cells. Well-known diabetes drug metformin works as an activator of AMPK. In pancreatic beta cells, INS1E, metformin inhibited TXNIP transcription, gene expression, and protein level in a concentration-dependent manner. AMPK exerts its effects on TXNIP by a carbohydrate response element binding protein ChREBP. They found that in the presence of lower glucose concentration, about 3.3 millimole per liter, ChREBP was mainly localized in the cytoplasm of the INS1E cells. But when the cells were incubated in high glucose, about 22.2 millimole per liter, in about 30 minutes, approximately 80% of the ChREBP was found inside the nucleus. They also reported that this high level of ChREBP inside the nucleus persists about 4 hours after stimulation with high glucose. ChREBP binding with DNA sequences contributes in the transcription of TXNIP. Glucose-induced TXNIP transcription depends on a 400 base pair proximal region of the TXNIP promoter, which contains a conserved ChO-RE consisting of 2 non-palindromic E boxes. Transfection experiments using luciferase reporter construct driven by the ChO-RE showed that high, about 22.2 millimole per liter glucose increased ChO-RE TXNIP promoter activity by about 5-fold relative to treatment with low glucose, about 5.5 millimole per liter, and metformin reduced the glucose stimulation of the show early TXNIP promoter. Zhao Liu Li et al. Published an article in August 2015 in the Journal of Molecular Endocrinology entitled New Insight into Metformin Action, Regulation of ChREBP and FOXO1 activities in endothelial cells. Dysfunction in vascular endothelial cells contribute greatly in diabetes related complications in different organs, especially in cardiovascular complications and fatalities. High glucose induced dysfunction in endothelial cells was found to be associated with the induction of TXNIP expression in primary human aortic endothelial cells. Both ChREBP and FOXO1 are dramatically recruited to the TXNIP promoter to activate strong expression of TXNIP upon high glucose. FOXO1 was also reported to repress the transcription of TXNIP in neurons. The action of FOXO1 on TXNIP transcription seems to be tissue and cell type specific. In aortic endothelial cells, metformin treatment suppresses high glucose-induced TXNIP expression at both messenger RNA and protein levels. TXNIP is now known as the most critical player of beta cell death in diabetes. TXNIP is also a great contributor in the pathogenesis and pathologies of other diabetes-related complications. Treatment of diabetes patients targeting TXNIP may give much more better outcomes someday in near future. I would like to wish good days for you everybody. Thank you.